transduction. Before we go in details of signal transduction, let's understand what are the objectives of this lecture, what we are going to study in today's lecture. The learning objectives that we are going to focus on is what is signal transduction, what is the process actually, what are the different steps in signal transduction. We are also going to talk about the receptors. Now, when we talk about signal transduction, it involves a lot of receptors and pathways based on those receptors. And we are going to talk about the total signal transduction process in short. And we are also going to see some of the uses of signal transduction in plants, like use in defense. So we are going to talk about all these aspects. And I hope at the end of this lecture, you will be able to understand all these points, and you will be able to focus on these points when once we have finished about the lecture. Okay, now let's go to the first point, the signal transduction or cellular internet. Now, when we talk about signal transduction, the word itself talks about, or word itself is an indication of something. Now, when you talk about transduction, do you know what is transduction? Anybody? Do you know what is transduction? Now, let me tell you what is transduction. Transduction is conversion of a message or energy from one form to another. Signal transduction basically talks about a signal that is being given to a cell. It is converted into a response, and this conversion of signal to response is called as signal transduction. So we are going to see how a simple signal will be transduced into a cell, and how this simple signal will be converted into a response, a visible response that might be a change in growth, that might be a change in shape, or that might be a change in a particular pattern. That is called as response. So how this a simple signal is converted into a response, that entire process is called as signal transduction. Now, when we talk about signal transduction, we must understand there is a cellular internet, just like the internet that we use, where many computers are interconnected with each other and they are constantly communicating with each other, or they have capacity to communicate with each other. Similarly, cells, they also have a internet or a communication network in them. This communication network sends signal and receives signal. Based on those signals, there are certain things that are done. That is why signal transduction is also many times referred to as the cellular internet. Now, what are the aspects of cellular internet or cellular communication? Now, if you see, cell-to-cell communication is an essential thing for multicellular organism, because there are many cells, there are many organs, and when an organism has many organs, these organs have to perform in a coordinated fashion. And when these organs have to perform in a coordinated fashion, there has to be a communication between all of these organs. Therefore, this is a very important process in multicellular organisms, where there are many tissues, many organs which are working together as a single organism. So in that case, cell-to-cell communication or communication between various structures becomes a very important phenomenon, and therefore signal transduction or cell-to-cell communication is a very important aspect in cell-to-cell or a cellular internet. Now, when you talk about signal transduction, it plays a very important role in perceiving the environmental conditions and adapting to these environmental conditions. Now, we know that we have organisms, they face environment, and this environment is constantly changing. Now, you must have seen the change recently, that a few days back the temperatures were very high, and now if you see currently or within next few days, the temperatures will go down, humidity will increase. Accordingly, there will be a lot of change in environment. Such change in environment has to be perceived by the organism. The organism la to change kadala hava, and once it has perceived that change in environment, that an organism has to adapt according to that environmental change. And for that purpose, signal transduction becomes a very important process, because of signal transduction, the organism is able to understand that there is a change in environmental conditions, and it has to adapt according to the environmental change. So this environmental change is perceived by these multicellular organisms, which includes plants also. The site of perception for plants is many times leaves, where the signal is perceived, and it initiates a cascade of events. These cascade of events or series of events will ultimately result into a response that will make that plant more adapted towards the changed environmental conditions. So signal transduction plays a very important role in environmental, sensing the environmental conditions and adapting according to the environmental conditions. When you see the signal transduction process and its study done, you will observe that biologists have discovered many universal mechanisms in cellular regulation or cellular communication. These mechanisms, they are having a common line right from bacteria to animals to plants. You'll find some common pathways or some common molecules that are involved in these pathways. It indicates that these pathways or these signal transduction methods or processes have evolved right from bacteria to up to the higher organisms such as plants and animals. So you'll find there's a common connecting link between all signal transduction pathways or all signal transduction processes, that they have certain receptors, they have certain processes, and they have certain responses. These are many times showing some similarity in the bacteria, plants, and animals. So when you see the entire research done on signal transduction, you will find that there are certain universal mechanisms that are working in signal transduction. We are going to talk about signal transduction in plants. So we are going to slightly focus on signal transduction in plants. Although a lot of information is available about signal transduction in animal, very scarce or limited information is available about the signal transduction in plant. The reason is very obvious. Plants don't have a nervous system. Animals have a nervous system, and they have well-developed endocrine system or system where hormones are produced in large number, and there are n number of animal hormones that interact and help in signal transduction in animals. Plants have a limited amount of hormones, and they have different types of signaling stimuli. Like plants have mechanical stimuli. When you touch a plant like Mimosa pudica or Lazaro, you'll notice that it shows some response to that. So there are certain different receptors, and their processes are different compared to animals. So we have some limited information about plants and their signal transduction, but we are going to focus on plants and we are going to focus on basic mechanisms, and we are going to talk about what is the specialty of plants, or what is the special thing that is observed in plants regarding that signal transduction process. So this is signal transduction. Now, when you talk about signal transduction and its evolution, you'll find that a signal transduction pathway is a series of steps by which signal is conveyed, or it is transferred from cell surface to interior of the cell. So the signal transduction many times starts with the surface of the cell, and it ends into a response inside the cell, which is many times visible to the observer. The signal transduction pathways, they are basically starting with surface molecules, and these surface molecules, they are basically receptors, and these receptors will start the process, and ultimately they will bring about certain changes, and that will lead to a cellular response. Now, if you analyze the signal transduction pathways, you'll see that the molecules that are involved in the signal transduction pathways and the various pathways, they are similar, or they have similarities right from prokaryotes to eukaryotes, and they have some adaptation as you go from prokaryotes to eukaryotes. Now, when you talk about eukaryotic signal transduction, the eukaryotic signal transduction pathways, they are slightly different in plants and animals. There are certain, receptors that are slightly different in plants. There are certain types of receptors that are predominantly seen in plants that are not seen in animals, or there are certain, receptors that are seen in animals and not seen in plants. So there is a difference in signal transduction pathways in plants as well as animals when you talk about eukaryotic signal transduction. Now, when you talk about signal transduction in plants, the basic outline of any signal transduction pathway that take place in plant has a particular, say, structure. Now, what is this structure? The signal transduction pathway starts with an external signaling factor. This external signaling factor can be environmental factor, which can be biotic factor, or which can be abiotic factor. Environmental factors such as light, temperature, or even the concentration of salts is an environmental factor, which will then other than this, you'll have biotic factors. We have biotic factors where we have the pathogens, we have many herbivores. Such signals or such external factors will lead to signal, and this signal or this particular process will lead to formation of signal, and this signal is perceived by plants. Now, perception of signal means understanding that certain change has occurred, and this understanding of the certain change is nothing but the perception of signal. This perception of signal is generally done by certain organs. In case of plants, many times this perception is done by leaves and other organs. Leaves are the major part of a plant, so many times you will find that many signals are perceived by the leaves. Now, in the leaves, especially on the cellular surface, you will find there are certain receptors and sensors. These receptors and sensors are the actual molecules which receive this signal. These receptors and sensors, once they have received the signal, they do the function of signal transduction. That means they convert this signal into a process. This signal transduction process or signal transduction is actually done, or it is executed by certain molecules which are secondary messengers, hormones, or signaling molecules which are produced in turn or which are produced in response to the signal. सिग्नल जो प्लांटला मिळतो त्या सिग्नलच्या रिस्पॉन्स मध्ये प्लांट प्रोड्यूस सर्टन मॉलिक्युल्स. दीज आर कॉल्ड एज सेकंडरी मेसेंजर्स, hormones, or signal molecules, signaling molecules, or growth factors. These molecules will further do or further transduce the message in the process. What they do, these secondary messengers, hormones, or signaling molecules, they bring about certain changes in the cell. What are these changes? These changes are like phosphorylation or dephosphorylation of certain molecules. These phosphorylation and dephosphorylation processes, they help in activation or deactivation of certain, say, key molecules such as the transcription factors. These, molecules which are key molecules which control a key process such as transcription or gene expression, they are phosphorylated or dephosphorylated. This phosphorylation or dephosphorylation activates or deactivates certain molecules. These activation and deactivation of these, transcription factors will lead to gene expression. Gene expression, it either will be positive or negative, means some genes will be upregulated or downregulated, or some will be completely stopped from expression. Now, what is upregulation and downregulation of a gene, or certain genes are stopped from expression? The term itself indicates a lot. Like upregulation of gene means these genes are overexpressed. These molecules which are produced from these genes are produced in higher amount. That leads to certain response. Or some genes are downregulated, means their expression is reduced, leading to less production of these molecules and ultimately producing certain response. So gene expression is controlled by phosphorylation and dephosphorylation of certain molecules. This gene expression will lead to transcription, mRNA formation, ultimately leading to formation of proteins and ultimately forming a physiological response in the cell. So the process starts with a phenomenon that can be a abiotic stress, or that can be a certain signal by light or temperature or by herbivores that will be perceived by the plants. The plants will produce a signal or secondary messenger. These secondary messengers will produce a reaction or produce a signal or a signal mediation reaction. This signal mediation reaction will include phosphorylation and dephosphorylation or certain changes in the key molecules. These key molecules can be transcription factors. These can be some enzymes which control a certain process. In case of transcription factors, when they are activated or deactivated, they will affect gene expression. Some genes will be upregulated, some genes will be downregulated, or some genes can be completely stopped from expression, or they will be their expression will be stopped, inhibited. Then if the genes are upregulated or downregulated, their transcription will take place, mRNA will be formed. From that mRNA, protein will be formed, and then that protein will ultimately bring about a physiological or cellular response in the plant cells. So this is a general outline of how signal transduction takes place in plants or any organism. This is the basic pathway which is followed by the organism, which starts with an external signaling and ends with a physiological or cellular response. So after understanding what is the outline of the signal transduction, let's go to the next point, that is how signal transduction takes place. Now, as we know, signal transduction starts with perception of signal and ends with the formation of a particular cellular response. It is not always that the signaling molecule is perceived by organ, and in that organ only the response is taking place. Many times this signaling molecule is perceived by one organ, and its action is somewhere else. Or many times plant produce a signaling molecule which is produced at one tissue and its function is at other. For example, plants produce hormones which are again signaling molecules which will signal the plant tissue to perform certain process. For example, plants produce hormones such as auxins. Now auxins, they are many times produced at the tip of shoots or shoot tips, and they have major function of root initiation. So their function is basically at roots, or when the hormone is synthesized at one place and its function is at other place, it has to be transported, and many times this transport takes place in the plant. This transport can be long distance transport, or that this transport can be a short distance transport. Now short distance transport is basically for that purpose when the hormone has, or the signaling molecule has function which is related to a tissue nearby. जवळच्याच टिश्यूला काहीतरी सिग्नल द्यायचा असतो. So in that case, this signal is transferred to a short distance, and this is called a short distance transport of signal. And sometimes the signal can be carried from one organ to other, and this is called as long distance transport. Animals, for example, जर तुम्ही animals मध्ये बघितलं तर you will see that animals have a lot of hormones. Most of the animal hormones are synthesized in pituitary gland, which is located in your brain as such. And if you see the hormone functions, they are at different organs like kidney or other organs. Now, these hormones which are synthesized in gland, and this pituitary gland, and this hormone is carried from one place to another. There the transport takes place through blood. Here in the plants, the transport takes place through the xylem and phloem, and you will notice that xylem and phloem play an important role in transporting the signaling molecules. Now this is long distance transport for plants from one organ to other. Sometimes the signal has to be transferred for a short distance from one tissue to other, and which is very close by. In such cases, for short distance transport, plant produce small molecules which can be volatile organic compounds. Even electric signaling can also be used for communication between two cells. Electric signaling means the membrane potential that plays a very important role. So membrane potential can transmit a signal from one cell to other. These are the examples of short distance communication between cells, and long distance communication can take place in plants. Specifically, long distance communication can take place through the xylem and phloem. Now there is one very particular example of long distance communication. Now you must have heard about animals. They can transfer their hormones from brain to various organs. Plants can also transfer their hormones from tissue to tissue. they can also transfer hormones, or they can signal plants which are far away from them. म्हणजे एका प्लांट मध्ये तयार झालेला हार्मोन हा दुसऱ्या थोडासा दूरवर असलेल्या प्लांटला सुद्धा स्टिम्युलेट करू शकतो, अँड इट कॅन गिव्ह और कॅन प्रोड्यूस रिस्पॉन्स इन दॅट प्लांट. याच एक फार सुंदर एक्झाम्पल माझ्या वाचण्यात आलं होतं की हाऊ प्लांट्स हार्मोन्स कॅन अफेक्ट other plants. A hormone produced by one plant can affect other plants, and how that can affect the entire ecosystem. The example is 1990s example. In Africa, we know that Africa is known for game reserves. That is, it has huge, say, forests, and where people come to visit and see different, say, animals, which include lions, which includes antelopes. So such a say game reserve was there, or safari was there, and the owner of the safari noticed that during particular summer season, a particular species of antelopes called as kudu is dying, and it's dying very fast and in very large number. And since the kudus are dying, the predators of this kudu, that is lions, they are also moving away from this say game reserve. And because of lack of lions and other animals, the safari was suffering, and there were no visitors to that safari. So what he did, the owner decided to investigate, and he hired a zoologist, Van Hoven, and he was given the task to find out why these kudus are dying and what is the effect, or what is the basic reason for that. When he analyzed the dead bodies of many kudus, he found that they had a very large swollen liver, and it showed a poisoning effect. And when it was investigated, it was found out that the poisoning was of tannins, and tannin poisoning is causing this say death of kudus, and ultimately that is leading to the loss for that say game safari owner. Now, when he investigated why these say kudus are dying of tannin, he found out that actually these kudus are killed by acacia tree. Acacia is killing all these trees. Now, when this report was put forth, everyone was surprised. What the explanation was? The explanation was very simple. During summer times, a lot of the herbaceous vegetation was not there. So these kudus were feeding on acacia tree. And acacia tree, when it is chewed upon, produces a signaling molecule called as ethylene. Now, this ethylene, which is produced, is a gaseous hormone, and it can be very easily carried with, say, the wind. Now, when during this, say, summer times leads to overfeeding or overgrazing of acacia trees by these antelopes. And these antelopes, when they were feeding on this acacia produced a high amount of these ethylene gases, and ethylene gases, their production was around 30 times more, 20 times more than the regular one. And this high amount of ethylene gas, which was carried away by wind, led to hypersensitization of other plants which were other acacia plants which were in the vicinity. When they got this signal that there is ethylene in the air, they started producing defense mechanism, or they started producing defense molecule. The prime defense molecule for these plants is production of tannins. They produce 30 times more tannins in the leaves, that lead to when these kudus, when they feed on them, that lead to poisoning, or that lead to harmful effect on their liver, and ultimately the kudus started dying. So this is an example how plants can signal that they are in danger, and these signaling molecules such as ethylene can be very easily carried to other plants, and it can be a very long distance say communication, and this will lead to say sensitization of other plants and bringing out change or effective change in the other plants also. So this is a good example of how signal transduction not only can work in short distance, long distance, but also in very long distance in case of plants, where their signals can not only affect the same plant, but they can also affect the other surrounding plants, and then they can bring about a certain response in that plant. So this was a very classical example of long distance signaling in plants, using various hormones, including the ethylene that was the prime example here in case of acacia. Now, this is a example of long distance, say, communication or long distance uh, distance travel or carrying the hormone to long distance. Now, after understanding this, let's talk about the signal transduction and various steps in the signal transduction. Now, when you talk about signal transduction, there are four important steps in signal transduction. The first step is reception of the signal. Second is this transduction of the signal. Third is the response, which is to be produced by the cell. And fourth is the termination of the signaling effect or signaling event. This is very important because if the signaling event continues, the same response will continue to take place, and that might not be necessary in all cases. So at the end of signaling process, the signaling event has to be terminated. So second is transduction of the signal. Third is response, which is very important. And fourth is the termination of the signaling effect. Now, we will see one by one what are these different, steps and how these steps are taking place in a signal transduction event. Now, the first step, now we have seen here, if you see carefully, there's a receptor. The receptor binds to the molecule, and this is called as the reception of signal. Now, these binding molecules are called as receptors, and there are several receptors in plants. We are going to talk about receptors in details when in the next part. So first thing that happens is the receptor binds to the signaling molecule. Second thing is the once the receptor has bound to the signaling molecule, the receptor undergoes the changes, conformational changes. It changes its shape and various aspects. That conformational change leads to relay of signal. That signal is relayed into the cell and in interior of the cell. This signal is relayed by various enzymes and various molecules. Now, there are certain enzymes which are capable of, say, transferring this signal to other molecules. For example, there are many kinases enzymes. Kinase is one of the very important enzyme that is seen in signal transduction. Kinase is an enzyme that transfers phosphate from one group to other. So kinases are basically those enzymes which will transfer the phosphate from ATP to another molecule. Now, once the molecule receives phosphate, many times that molecule which receives the phosphate becomes activated and does a certain function and performs a certain function. And once the phosphate is removed, it again becomes inactivated and the function is stopped. So in this process, in the transduction process, many times the signal is transferred from one molecule to other via some enzymes like the kinase enzyme, which transfer the phosphate from one molecule to other. And this transfer of phosphate will lead to activation of certain molecule. These molecules will perform a task and then again get deactivated. The performance of task will lead to activation of another molecule. This activation of other molecule will lead to activation of next molecule. Like this, there is a chain of events, and that chain of event is called as transduction of the signal. That signal which is which has started from the reception of the signal or from the receptor, that will be transduced by various components, various molecules, various enzymes from one to other, and in a series manner this signal will be transferred. Ultimately, the signal is reached to a response molecule and ultimately leads to formation of a cellular response. This cellular response can be change in gene expression. This can be change in cell shape or change in division pattern. This can be any response that depends on the cell signaling pathway. So there are three basic steps: the reception, transduction, and response. Reception involves a receptor. Transduction involves many molecules which are also known as secondary messengers. These secondary messengers will pass on this message from one molecule to other, ultimately leading to the third step called as response, where the final process, that is activation or deactivation or upregulation or downregulation of certain genes will take place. or even in some cases the change of developmental pattern will take place, or change in shape of cell will take place, or any other cell division pattern will change, or cell division will start, or it will be initiated or stop. Depending upon what kind of pathway is, the activation response will be there. So the third part will be response, and final step, which I have mentioned here, is the terminalization of the signaling event. That is, the signaling event is ultimately stopped. You can imagine if this signaling is for cell division, and if the signaling event continues, it will lead to uncontrolled cell division and ultimately will lead to cancerous growth. So this signal has to be stopped when the desired effect is obtained. So many times terminalization of signaling event takes place. This terminalization of signaling event takes place either by positive or negative feedback control, where certain molecules from this chain of molecules will act as a feedback control and will lead to say reset of the receptor. The receptor will say release the signaling molecule, and it will be reset for receiving another signal. So this is the last step called as terminalization of signaling event, which is also very important. If the terminalization of the signaling event does not take place, the signal can remain as it is and can lead to some disastrous effect, especially in animal cases where it can lead to cases like cancer formation. So in case of cell division, of course. So these are the four important steps. Now we are going to talk about each step, and we are going to see details of each step or each phase as such. This is the third step, or the entire steps in a glance. You can see here, this is the plant cell showing various events, which includes a perception of signal. Now this is the cell wall. Inside it, you can see there the plasma membrane, and there are molecules which can perceive signal. You can see here, this is a phytochrome that is perceiving signal of light. This is one more channel which can receive signal of certain molecule, or even light can trigger these channels. These will ultimately lead to formation of a signal, and this signal will be transduced into the cell. You can see there are molecules formed. This is cyclic GMP which is formed, which is nothing but a secondary messenger. Here calcium ions which are released are formed in this in the cytoplasm. They act as a secondary messenger, and these GMP or calcium ions, they can activate certain kinases, and these kinases will ultimately go inside the nucleus, and they can activate certain transcription factors, and these transcription factors will ultimately control, either up regulate, down regulate, or completely stop the expression of certain genes. This is an example of deotylation or greening of leaves. This is the example from that. So you can see the response given here is the deetylation of the leaf. So such response can be obtained by a simple reception of signal. So this is an example of the entire process: signaling, signal reception, production of secondary messenger, and from secondary messenger, this is also a messenger molecule, that is a kinase enzyme, which will go inside and say activate certain key molecules such as transcription factor, and that transcription factor will bind to the gene and will either upregulate or downregulate the gene expression. So this is the actual process, the three processes together. Let's go to the first step, that is reception of signal. Now, signal is generally received by a receptor protein. Now, these receptors are many times two types, that is membrane receptors and intracellular receptors. Now, membrane receptors, as the name indicates, they are present on membrane, and these membrane receptors are basically hydrophilic in nature. They can interact with hydrophilic molecules, and they will bind with molecules which are say from the environment, and once they are bound to the molecules, they will undergo, these receptors will undergo changes, and these changes will lead to formation of, or the transduction of say the signal, and ultimately leading to effector molecules, which can be transcription factors, which can be metabolic enzymes, which can be cytoskeletal proteins, and ultimately bringing about the desired result, that is altered metabolism, altered gene expression or altered shape of the cell. So basically, there are receptors which are basically of two types. One is the membrane receptors. The other is the intracellular receptors. Membrane receptors, as I have told you, that these are hydrophilic in nature and they bind to the water-soluble substances. Then there are second type of receptors, which are the intracellular receptors. And these intracellular receptors, these are present in the cell, inside the cell, and they bind to hydrophobic molecules. Now, when we talk about signaling molecules, there are two types of signaling molecules that are observed commonly. One is the hydrophilic signaling molecules or signaling ligands, and hydrophobic ligands. Signaling ligands or signaling molecules can be hydrophilic and hydrophobic. Basically, hydrophilic means soluble in water. Hydrophobic means not soluble in water, and many times they are soluble in fat, or they are soluble in lipids. Now, lipid soluble and water soluble ligands, and based on these two, there are two types of receptors. The water soluble ligands are binding to the membrane receptors, which are present on membrane, whereas these hydrophobic lipid soluble molecules, they are binding to the intracellular receptors that are present inside the cell. Can anybody tell why these hydrophobic chemical messenger or hydrophobic ligands can directly go inside and bind to the intracellular molecules or intracellular receptors inside the cell? What can be the reason, or how it is possible? Whereas the hydrophilic molecules, they cannot cross the membrane. They have to bind to these membrane receptors. Any idea? Okay. The reason is very simple. These hydrophobic messengers or these ligands, they are fat soluble. So they are very easily capable of passing through the plasma membrane. We know that plasma membrane is lipoproteinous, made up of lipids and proteins. So since these molecules are lipid soluble, they can easily cross the barrier of the plasma membrane and go inside. And once they are inside, they can be bound by these intracellular say receptors. Now, one more thing that we should understand here is there are certain molecules which might not be fat soluble, but they are transferred across the membrane and they have intracellular receptors. For example, auxin is transferred through transferred through the membrane by a transporter called as Lux transporter. This Lux transporter transports the auxin from outside the cell into the cell, and because of that, the auxin is having a intracellular receptor also. So receptors are two types: a membrane receptor, which is binding to many times binding to the hydrophilic ligands, and then there are intracellular receptors which are binding to the many times hydrophobic messengers, and which will come directly inside the cell, and these molecules inside the cell will bind to the intracellular receptors. There are some exceptions, like the molecules such as auxin will be transported through the membrane transporter such as Lux transporter, and this transported molecule can bind to the intracellular receptor inside the cell. Okay. So receptors we have seen. Now let's talk about the different types of receptors based on their functions as such. Now most of the water soluble, that is membrane receptors, there are 4 types of membrane receptors that are there. Basically 3 types. One membrane receptor is which is a photoreceptor, can be present inside the cell also. What are the 3 types of main say membrane receptors? These membrane receptors are G protein linked receptors, receptors which are tyrosine kinase or enzyme linked receptors. Third is ion channel linked receptors, and fourth receptor that I have mentioned here is the photoreceptor that is present in the cell. The other type of receptors, that is intracellular receptors, these are receptors for auxins, jasmonic acid, gibberellin, which are present inside the cell and which can bind to the molecules inside the cell. So these receptors are present inside the cell, whereas G protein linked receptor, enzyme linked receptor and channel receptors, they are present on the membrane. So membrane receptors and intracellular receptors. The membrane receptors are G protein linked receptors, enzyme linked receptors, and channel receptors. And inside the cell, intracellular receptors, we'll have several receptors which can bind to molecules such as auxins, jasmonic acid, and gibberellin. We'll see one by one what are these three types of receptors on membrane, and we will also briefly talk about the intracellular receptor. This receptors on the cell, you can see these are the three main surface receptors: ion channel linked receptors, enzyme linked receptors, and G protein linked receptors. Now, if you see, as the name indicates, ion channel receptors are the receptors which are actually ion channels. And when a ligand binds to the ion channel receptor, the ion channel opens and will facilitate entry of certain molecules inside the cell. So this is an ion channel receptor. Whenever the ligand dissociates, the channel is closed, the entry is prohibited. Next is G protein coupled receptors, or also known as GPCR. These GPCR or G protein coupled receptors, these are present on the membrane. They are known as G protein coupled receptors because they have a protein linked to it which has GDP attached to it. That is why it is called a G protein linked receptors. And third kind is the enzyme linked receptors, where the receptor has extracellular domain which has ligand binding site, and intracellular domain which has enzymes such as tyrosine kinase enzyme, and which can perform kinase activity. So these receptors are linked to enzyme, that is kinase enzyme, and once the ligand binds to the receptor, these enzyme linked receptors will activate these enzymes such as tyrosine kinase enzyme. This tyrosine kinase enzyme will transfer phosphate from ATP to a certain molecule that contains tyrosine, and ultimately will phosphorylate that molecule. That molecule, once got phosphorylated, becomes activated and then will perform certain function in the cell. So these are the three main receptors that are present, and they play a very important role in the cell signaling. Now, this diagram shows the different types of receptors that are in plant. The same point is again shown here. You can see here there are receptors. For example, this is a brasinosteroid receptor. This is an abscisic acid receptor. This is a mechanical receptor that will sense the mechanical or sense of touch. Then there are receptors for cytokinin. There are receptors for ethylene. Then there are receptors inside the cell. You can see these are the receptors which are present inside the nucleus. These are COL1 or TIR1. These receptors are actually the intracellular receptors which will bind to the molecule directly. For example, this TIR1 is a receptor for IA. This IAA molecule will directly come and bind to this receptor, and this will bring about changes in the receptor, and that will ultimately bring about changes in other molecules, and ultimately lead to up regulation or down regulation of genes in the nucleus. So this is an example of intracellular receptors like the TIR1, COL1, or even GID1 for gibberellin. These are the receptors which are present inside the cell. These are called as intracellular receptors, which can bind directly to the molecule which is transported from outside to inside the cell. Once inside the cell, these receptors will bind to these say molecules and bring about the desired say signal transduction. So these are some of the examples of say receptors on the plant cell. Let's go to the first type of receptor, that is ion channel linked receptor. So we have seen three types of receptor: ion channel linked receptor, second the enzyme linked receptor, and third is G protein receptor. We are going to see one by one. First is ion channel linked receptor. The ion channel linked receptor is a very important receptor. The receptor shown here is GLR receptor, that is glutamate receptor like protein receptor. Now you can see here this is GLR protein present here. Now here you can see there is a long chain of protein, or that is having N terminal outside, that is extracellular, and C terminal is inside the cell. And you can see there is a domain here which acts as a binding site. This GLR or glutamate receptor like protein binds to the glutamate amino acid. And once it has bound to the glutamate amino acid, it opens the channel, that is the calcium channel. This channel is actually a calcium channel that allows the entry of calcium inside. Now, once the receptor has been bound with the amino acid, it leads to production of secondary messenger, that is calcium here. This calcium will bring about a lot of changes. You can see what are the different changes that this calcium can bring about. It can bring about hemostasis of calcium that leads to pollen germination. It can also bring about the ROS production that can lead to closure of stomata. It can lead to, say, inhibition of the abscisic acid pathways that leads to seed germination. It can also alter cell division and the rate of cell division. So calcium ions, which are the secondary messengers that are produced after the binding of the amino acid glutamate to the GLR receptor, opens the calcium ion channel, and that channel, once it is opened, calcium ion concentration inside the cell increases. This secondary messenger calcium can bring about a lot of changes, which includes pollen germination, closure of stomata, seed germination. Even it can control the cell division in the root apex. So such example, this is an example of ion linked or ion channel linked receptor. So this is the first kind of receptor that that is there in the signal transduction, that is ion channel linked receptor. I hope this is clear to everyone. Let's go to the next type of receptor. This is G protein coupled receptor. Now, as I have told you, this is a receptor that is coupled with a protein that has GDP attached to it. This GDP controls the function of the protein. Before we go to the GDP or G protein that is attached to this, let's see what is this receptor. Now, if you see the structure of receptor, this is the actual receptor. This is also called as 7-spanning receptor. This 7-span receptor, why it is called a 7-span receptor? If you carefully see this receptor, you'll notice that there are alpha helices. These helical structures, these are, if you count them, these are 7 in number. 1, 2, 3, 4, 5, 6, 7 helical structures. These are transmembrane structures, and these transmembrane structures are present in this particular G protein coupled receptors, and these are called as 7-spanning proteins. They have a binding site here, and to this binding site the ligand binds or attaches, and once the ligand is attached to this binding site, it brings about the desired change. This is the first part of the G protein linked receptor. This is the receptor as such, and if you see, to this receptor there is another say protein attached called as G protein. Now, what is this G protein? Now, here this diagram shows this is the receptor, which is 7-spanning receptor, and to this, there is very close to this is one more protein called as G protein. This G protein is a heterotrimer, means it has 3 different say subunits. These are 3 different subunits with the different say size and credentials. If you see there are 3 alpha, beta, and gamma units, subunits. Out of this, the alpha has a GDP attached to it. This GDP has a 2-phosphate diphosphate molecule. Now, when the receptor binds to the the ligand binds to the receptor, you will see that what happens is, once the ligand has bound to the receptor, this GDP gets replaced with GTP. What happens is, this GDP is removed, and you get formation of GTP here. Now, once the alpha subunit has GTP, alpha subunit becomes activated. This alpha subunit of this G protein detaches itself from this G protein and goes to another enzyme. These enzymes can be an effector enzyme or an enzyme in the series of various molecules involved in the signal transduction. So what happens is, this alpha subunit, once it has a triphosphate attached to it, it goes and binds to another enzyme. This enzyme, once it receives the alpha subunit, what it does is, it also gets phosphorylated. You can see here it gets activated, and once it is activated, what it does is, it starts performing. Here you can see the enzyme here is adenine cyclase. enzyme that is used to convert ATP into AMP, that is cyclic AMP. Adenine cyclase has a very important function of converting ATP into AMP, cyclic AMP. And cyclic AMP, just like calcium, calcium plays a very important role. So this beta alpha subunit, once it has say detached, it goes and activates another enzyme called as adenylene cyclase. This adenylene cyclase converts ATP into cyclic AMP. This cyclic AMP, just like calcium, is a secondary messenger, and that can bring about a lot of changes in the cell and ultimately bring about the desired change. So or desired response. So this, once the adenylene cyclase is formed, what happens is the GTP is now converted to GDP now, and the GDP, once it is GDP, the alpha subunit becomes inactivated now, and that goes back to its original position. And now the system is reset. It will start or it will wait for the next say ligand to bind. Once the GDP is back, this ligand will also dissociate, and this receptor will also be ready for next ligand to bind. So this is a G protein or G protein coupled receptor that plays a very important role. Now, in plants especially, this kind of receptor is very rare. You don't find G protein receptor in plants many times. You will find that the next type of receptor which is there is very common in plants. But this type of receptor, that is G protein receptor, are very less or less in number in plants compared to animals. Animals, you will find G protein coupled receptors are more in number, and they are more abundantly found in the signal transduction pathway. In plants, you will notice that the first one, that is the ion channel coupled receptors, and the third one, that is the enzyme linked receptors, are the most common. These G protein coupled receptors are very rare or very less in plant signal transduction, but they are there. That is why I have included this in the presentation. So this is G protein coupled receptors. This is a simple say animation that shows G protein
coupled receptors, or GPCRs, are cell surface receptors that associate with heterotrimeric G proteins and mediate cellular response to signals such as light, hormones, cytokines, and neurotransmitters. All GPCRs contain 7 transmembrane alpha helices separated.
So this is G protein, which includes all G proteins. They have this receptor, which is 7 transmembrane alpha helices. And you will notice that this G protein initially has GDP attached to it. Once the signal is received, it becomes GTP, and then the signal can be further transferred. The G protein coupled receptors can be activated by light, can be activated by hormones, can be activated by certain molecules, and that is generally very common in animals and very rarely seen in plants. So after G protein coupled receptor, the third type of receptor, this is an example of G protein receptor, where you can see here there is a G protein here, many receptors which are light receptors. You can see here, this is the phytochrome, where the PR gets converted to PFR, then it activates the G protein. G protein activates the cyclic cyclic nucleotide formation. Now, in case of animals, there is adenine cyclase, which produces cyclic AMP. In case of plants, whenever G protein is present, there is guanylate cyclase. This guanylate cyclase produces cyclic GMP. You can see here there is cyclic GMP produced. This cyclic GMP acts as a secondary messenger, and that can lead to light related gene response, or that leads to response as such. So this is an example of G protein present in plants, where these phytochromes, which will receive the signal, will transmit that signal from PFR to G protein. G protein will produce cyclic GMP, and this cyclic GMP will regulate the gene expression and ultimately bring about the response that is required. So this is a rare example of G protein present in plants. Very few examples or very few processes involve G protein in plants. The next important receptor in plants is enzyme linked receptors. Enzyme linked receptors, you can see there is an array of receptors given here. These enzyme linked receptors have 3 domains. One is the extracellular domain, other is the transmembrane domain, and the third one is the intracellular domain. 3 domains are there. The extracellular domain is for binding of ligand. The transmembrane domain is for anchoring the receptor, and the intracellular domain, which is generally an enzyme, which is a kinase enzyme, is also called as kinase domain, is also present. So three domains. Now these enzymes or these receptors, they are present on the cell, and they are linked to kinase enzyme. Now there are several receptors that are present in plant, or which are very common in plants. For example, this LRRRK receptor, which is shown here. This LRRRK receptor is leucine rich repeat receptor kinase. It is LRRRK, means leucine rich repeat receptor kinase. This here LRRRP is leucine rich repeat receptor protein. So these, if you see carefully, there are small things or small structures which are repeated constantly. These small structures which are repeated are leucine rich motifs, or leucine rich areas of the protein, and they are present on the receptor. That is why it is called as leucine rich repeat receptor kinase, or leucine rich repeat receptor protein. So these leucine rich receptors that are present. Along with this, you will also find there are lectin rich receptors. Then there are EGF, that is epidermal growth factor rich kinases, or there are lysine motif rich kinases. So these are some of the enzyme linked receptors. You'll find that if you see all of these enzyme or these receptors, you'll find that they have something in common. That is, they have three parts: the outer or the extracellular part, the transmembrane part, and the intracellular part. The extracellular part is for binding of the receptor to the ligand. The transmembrane part is basically the transmembrane part is basically for anchoring the receptor, and the inside the cell, the part that present is the kinase domain. This kinase domain is basically an enzyme, the kinase enzyme, that can transfer phosphate from one molecule to other. So this is enzyme linked receptors. Now, whenever the enzyme linked receptor, now you can see here these enzyme linked receptors, they all have kinases. So these kinases, they have an activity of transferring phosphate from one molecule to other. Basically, they use phosphate from ATP to transfer it on the other molecules. And these other molecules are also enzymes. They get activated, and once they are activated, they transfer the signal from one molecule to other. Now, if you see the mode of action of these enzyme linked receptors, you will see that here the diagram shows how the enzyme linked receptors amplify the signal. Whenever there is a signal, you can see here the signal is perceived. The signal leads to formation of secondary messenger, or it leads to formation of or phosphorylation of certain molecules. Now here the molecule shown here is MAP3K, that is mitogen activated protein kinase 3. These mitogen activated protein kinases, these are enzymes which can act as kinases and transfer phosphate from one molecule to other. So whenever the signal is received, these enzyme linked receptors phosphorylize the MAPK or MAP3K. This MAP3K in turn phosphorylates the MAP2K. The MAP2K in turn phosphorylates the MAP kinase, and ultimately this MAP kinase phosphorylates the target protein, and the target protein will do the function or will perform the response as such. Now this is called as signal amplification, because at every stage when there is a phosphorylation step taking place, there are more molecules produced. That is, initially the MAP3K molecules are less compared to the MAP2K. So the MAP3K will activate more molecules of MAP2K. MAP2K will activate more molecules of MAPK. So ultimately MAPK molecules will be large in number compared to the initial starting number of MAP3K. So these signaling molecules which are produced at the K, that is MAPK level, that is the last level, they are very high in number, and they produce a very strong, say, response. So at every level where the signal is transferred, there is amplification of signal, because at every level there are more signaling molecule produced. In initial stage map 3K, the numbers is less, but at map 2K the number increases, the map 2K number increases, map 3K number will be more larger than map 2K number, and ultimately that will activate large number of target protein, and those large number of target protein will bring about the desired response. So at every level, when this kind of say enzyme linked receptors are there, they transfer the signal, and at every level you will find that there is amplification of signal, and you will get a very strong signal form. So these are the three types of say signaling receptors. After understanding the three receptors, the next point that we should understand in the signal transduction pathway is the secondary messenger. Now we have discussed there are two secondary messengers that we have seen: cyclic AMP and calcium ions. Calcium ions are one of the very important secondary messengers in plants. Cyclic AMP also plays an important role, but it is more involved in the say signal transduction in plants, animals and bacteria. But this calcium plays a very important role in plants. So secondary messengers are basically those molecules which are activated by the receptors, and these molecules, they receive the signal from the receptor. रिसेप्टर कडून मिळणारा सिग्नल किंवा रिसेप्टर जो ज्या मॉलिक्यूलला सिग्नल देतो, जो दुसरा मॉलिक्यूल असतो त्याला सेकंडरी मेसेंजर म्हणतात. And many times this secondary messenger in plants is calcium ions. We'll see some examples of production of secondary messengers and how they work. Now there are two examples. This is one more example of secondary messenger. I have told you that the secondary messenger is calcium is the secondary messenger. Now here this is a secondary messenger production, which is production of say calcium ions or release of calcium ions in the cytoplasm. This takes place when a ligand binds to a messenger. The messenger ligand binds to the receptor, and once the ligand binds to the receptor, it activates this is a G protein or G protein coupled receptor. This G protein coupled receptor, once it has bound with the ligand, the alpha unit of the G protein gets converted to active form by replacement of GDP by GTP. That will activate a lipase enzyme. This lipase enzyme will convert phosphatidyl inositol 2-phosphate into phosphatidyl inositol 3-phosphate or IP2, PIP2 to IP3. This IP3 is another molecule. This IP3 molecule goes and binds to the calcium channel on the endoplasmic reticulum, and that will release lot of calcium, and the calcium acts as the secondary messenger. So here actually IP3 also is a secondary messenger, but more stronger secondary messenger is the calcium ions, which will produce a lot of changes in the cell as such. This is one example of the secondary messenger production by G protein coupled receptor. The other example here is the same one that we have discussed earlier, that the ion channel coupled receptors. You can see here the receptor binds to the amino acid, that is glutamate, and that increases the cellular concentration of calcium. Once the calcium is inside, it will bring about a lot of changes. Now, what are the changes that calcium bring about? You can see here the calcium ions play a very important role. You can see here, once the calcium ion is inside, it can activate, deactivate a lot of things. For example, the calcium ion, once they are here, you can see this is the pump that is allowing the calcium ions to come. This is a say channel related or channel coupled receptor, which is allowing the calcium ions to come in. Once the calcium ions are inside, they bind to various molecules such as CDPK. This is a kinase enzyme. This kinase enzyme will activate other molecules, other enzymes. These other enzymes will activate certain transmembrane transporters, which will transport certain molecules inside or outside. They can also activate pH pumps or OH or H ion pumps which will alter the pH of the cell, or these calcium ions can bind to certain molecules such as calmodulin. This is a molecule called as calmodulin. This calmodulin is a protein molecule which can bind to calcium. Once it binds to calcium, it can go inside the nucleus and bind to transcription factors. And these transcription factors can ultimately bring about the change in the gene expression. So there are several roles that this calcium ions can play. For example, they can activate certain pumps on the vacuole, which will pump out a certain molecule inside or pump out certain molecule outside. Based on what kind of pumps they get activated, the entire cellular osmoticum can be changed. The calcium ions, hydrogen ions concentration can be changed. Once hydrogen ion concentration can change, the pH of the entire cell can change. So these are some of the very important changes this secondary messenger such as calcium can bring about. So we have seen different types of receptors. We have seen how the receptors bind, and now we have seen the secondary messengers, how these secondary messengers, they bring about the changes. And now, after understanding the secondary messengers, let's talk about the last step, that is a response. Response in the cell can be various. So what are these responses? Now you can see here, this is a diagram showing various stimuli, and based on that, various secondary messengers and their responses. Now, for example, here you can see a pathogen is trying to enter the cell. So it is degrading the cell wall. It produces a signal that leads to formation of various molecules, which are nothing but the secondary messengers, which includes ROS, which includes calcium ions, which include change in pH, certain lipids, certain sugars are formed, and certain ion concentration also changes. Based on this, various responses can take place. It can lead to ROS accumulation, that can lead to change in the transcription, and ultimately that can lead to a gene expression changes. And ultimately, once the gene expression changes, it can lead to extra deposition of cellulose and lignin, and that will prevent the entry of pathogen inside the cell. So you can see here the pathogen tried to enter, producing a secondary response or secondary messenger. So the secondary messenger that are produced, they produce or they activate certain molecules such as MAP. The MAP molecules can activate transcription factors. The transcription factors can then alter the gene expression and can lead to extra callose and lignin deposition on the cell, leading to protection of the cell. Certain say stresses like abiotic stresses will also produce secondary messengers. These secondary messengers will also carry out certain processes by which the message will be carried, and ultimately a response will be given. So this is an example of how different stimuli can bring about a complex signal transduction and ultimately lead to formation of a response. So this is an example of formation of a response. You can see there are several things that occur in a cell in response to various stimuli, which can be sugar accumulation, which can be lipid peroxidation, which can be extra deposition of callose and certain molecules on the cell wall. These are some of the changes that can be seen as a response in the cell after signal transduction. This is one more example of signal transduction in case of auxin. Auxin, where the regulation of gene control done by auxin. You can see here, this is a transcription factor which is controlling the auxin regulated genes. That is, these genes are controlled by auxin. It has a transcription factor which controls the expression of the gene, but to this transcription factor there is an inhibitor attached. This inhibitor will only be removed when auxin comes and binds to it. Now, when auxin comes and binds to this inhibitor, once the inhibitor is bound by auxin, protein degrader, or also called as the ubiquitin complex, which will bind to the auxin inhibitor or auxin gene inhibitor. And once the receptor is once the molecule is bound, then there will be degradation of that molecule, inhibitor molecule. And once the degradation has taken place, the transcription factor will be able to function, and the auxin related gene we stay expressed.
इकडे बघ.
Once the oxygen-related genes, there are certain changes that take place. So finally, one more response that is I'm going to discuss today is the responsible auxins. We know that auxins are responsible for cell elongation. How this cell elongation takes place? The auxins, once they are inside the cell, they lead to certain gene expression. Just now I have shown you that these auxins, once they are inside the cell, they can say regulate gene expression. This is the auxin gene expression where they can upregulate this gene expression. Auxin regulated genes, they are upregulated. Once they are upregulated, they form certain protein channels. These protein channels, once they are formed, they are incorporated in the plasma membrane. The plasma membrane मध्ये जातात, and they start pumping H plus ions into the into the cell wall. Now, once the H plus ions are into the cell wall, that lowers the pH, that makes the cell wall acidic. This acidic cell wall will activate certain molecules called as expansins. These expansin molecules will lose or loosen the various fibers present, that is cellulose fibers present, microfibers of cellulose that are present, or cross-linking polysaccharides that are present. They are cut or they are made loose. And once the cell wall becomes loose, it is very easy for the cell to expand as the water is entered into the cell and the cell becomes turgid. Since the cell wall is loose because of the H plus ions, the cell very easily expands. So this is how the cell expands with the help of say the auxin, and how the auxin brings about the response. Auxins come into the cell, binds to the transcription factor. Transcription factor leads to expression of certain genes. These genes are basically proton pumps genes, and these proton pumps that are formed, they go into the the plasma membrane, incorporate into the plasma membrane. These proton pumps will pump more hydrogen into the cell wall. As the cell wall gets more hydrogen, it becomes acidic. As the cell wall becomes acidic, it affects or activates expansin. These expansins will cut or loosen the cell wall microfibers of cellulose, and that will make the cell wall loose. Once the cell receives water or endoosmosis takes place, it becomes turgid. As the cell becomes turgid, since the cell wall is loose, it can expand and you can get cell elongation because of auxin. So this is how actually a response takes place by signal transduction. Auxin to nucleus where it affects the transcription factors. The transcription factor leads to upregulation of genes. Upregulation of genes leads to formation of more H ions transport or proton ion transporters. These proton ion transporters leads to acidic cell wall. Acidic cell wall leads to expansin activation. Expansin activation leads to loosening of cell wall, and loosening of cell wall leads to the elongation of cell. So this is how the cell signal of auxin is transduced, and ultimately response is brought about. Finally, the termination of the cell signal. Once we have seen the receptors, we have seen the secondary messengers, we have seen an example of how this messenger or how the process takes place in case of auxins. Finally, we'll talk about termination of the signaling mechanism. The signaling mechanism can be terminated by positive feedback or negative feedback mechanism. Here, a positive feedback mechanism where a signal comes, it activates the mechanism, and final molecule which is formed will again activate or will again strongly activate the the process. This is called as positive feedback. But this positive feedback is not used for signal termination. Signal termination is done by many times by negative feedback mechanism, where the signal leads to formation of the final response. The response molecule will again go and stop the signaling molecule or reset the signaling receptor, and ultimately this process will be stopped. So the final product will have a negative effect of the entire process, and ultimately that will bring about the stoppage of signaling process. And there is one more mixed process where positive feedback and negative feedback both work together. where there are 3 steps. Signaling leads to formation of a secondary messenger. Secondary messenger will again have a positive effect on the process and lead to more production of secondary messenger. More secondary messenger will lead to final product, that is C. And this C, when it is reached to a threshold level, एका threshold level ला गेल्यानंतर C will start inducing negative feedback on A, and ultimately the process will stop. This kind of many times this kind of say signaling mechanism or termination mechanism is seen, where the intermediate has a strong positive effect, but the final product has a negative effect in the chain of events. So this is termination of the events that takes place, signaling events that takes place many times in the signaling pathway. Finally, I will just like to shed a light on how these say signaling pathways help in the plants. There are many processes that are helped by the signaling pathway. There are 2 processes that I would like to focus here: hypersensitivity response and acquired resistance response. These are produced by pathogens. This is very commonly seen in the environment, where you can see that pathogens, they start attacking a plant. When they attack a plant, they produce certain signaling molecules. These signaling molecules, they lead to formation of a hypersensitive response in the plant. These hypersensitive responses many time leads to formation of antimicrobial molecules, then sealing of the cell wall, which are of the cells which are already infected by the microbe or by the pathogen, and ultimately leading to formation of methyl salicylic acid. This methyl salicylic acid, which is produced in the cell which is attacked, this methyl salicylic acid is then transferred to other cells, where it gets converted to salicylic acid, and this salicylic acid acts as a signaling molecule and leads to acquired resistance. That is, it leads to formation of certain molecules which will give resistance against that particular say pathogen, invading pathogen, and the plant will be ready for that particular pathogen. So this is another example of how cell signaling helps in help plants in defense and protecting itself from various say microorganisms. So this is an example of how signal transduction is used by the plants. So this is the final slide. Thank you very much for patience and say calm listening from all of you. Thank you. Thank you very much.